Today I'm going to talk about Filamin A and how it regulates platelet genesis and platelet life, and how it regulates its biogenesis. Okay, so in my talk, first I'm going to talk about platelets in general and how platelets are being formed. Then I'm going to talk about the role of Filamin A in platelets genesis and life, as I said. And in the final part, I'm going to show some of still unpublished data on Filamin A and control of thrombopoietin receptor. So for the beginning, so as probably all of you know, platelets are the smallest of blood cells, and when they were first observed in the 19th century, they were described as dust of blood, and people are not sure if they are really derived from blood or they come out from some other sources from environment. And it was actually Italian researcher Begonja from University of Pavia that was the first to characterize them, to characterize their morphology and to actually describe what they do, that they are important in hemostasis. So they are very small cells, 2 to 4 microns, and as you can see here on these electron micrographs, what characterizes them, that these are cells without nuclei. Well, to be more precise, mammalian platelets do not have nuclei, but if we go to birds, fish, or reptiles, they have thrombocytes, which do have nuclei and which develop directly from progenitor cells. So mammalian platelets, or here human platelet, as you can see, it is rich of granules. It is full of granules called alpha granules or dense granules, depending on the how it breaks the light, and full of mitochondria. And as platelets are discs in circulation, on the side section by electron microscope, you can see how they are rich also in internal membranes. These membranes are called open canalicular system. It's a system that is connected with the surface of platelets, and these membranes will serve platelets to increase their size once it needs to be activated. So the major role of platelets is that they recognize vascular wall damage. So when they are in resting state, they are discs, but once they see extracellular matrix proteins in your vessels, they will start to be activated and they will make aggregates, and this is how they will stop bleeding. In recent years, there has been many discoveries or studies on other roles of platelets. So platelets have been shown to be important as important factors in development of lymphatics. They also have been recognized as important cells of innate immunity, since they express many TLR receptors and they secrete some bactericidal proteins. So some of the clinical facts. So in our circulation, there's about 10 to the 12 platelets. Their lifespan is 8 to 10 days, so quite short, let's say. In mice, it's even shorter. It's about 5 to 6 days. And every day, our body produces 100 billion new platelets, which is quite a big task to maintain the normal range of platelets in blood, between 150 to 40 platelets 10 to the 8 per milliliter. So there are clinical conditions in which high platelet numbers will lead to increased risk of thrombosis, and on the other hand, there are clinical conditions with very low platelet numbers that will lead to increased risk of bleedings. So transfusion of platelets are in some cases the only therapy possible, and the only possible source of these platelets are donors that will donate blood. So it is very important to study molecular mechanism of how platelets are being made in order maybe in future to make in vitro platelets ready for transfusions. So in this video, maybe we can appreciate how platelets actually work. I will just start with. So these are human platelets isolated from whole blood, and we coated a surface with a trap. This is agonist of platelets, and you can see when platelets that are disc first makes contacts with the surface, they become spherical. They start to make these finger-like projections. Whole platelet is being flattened. Their surface increases, and they really seal and cover the surface, making this broad lamellipodia. These are really like band-aids, and you can imagine how they really cover the vascular injuries very nicely. So these are quite remarkable cells, although they do not have nuclei. So on the next topic, how platelets are really being formed. So platelets derived from one of the biggest cells in our body, and these are megakaryocytes in the bone marrow. Their size can range from 50 microns to 100 microns. And megakaryocytes develop from hematopoietic stem cells in a process called megakaryopoiesis. So this process roughly can be divided into 3 parts. So in the first part, it starts with a commitment of hematopoietic stem cells as a response to certain stimuli to differentiate into megakaryocytic lineage. The most important stimuli or cytokine that drives this differentiation is thrombopoietin, which I will talk about later. So in this first step, what happens is proliferation and the formation of a pool of megakaryocyte progenitors, which will then, in a second step, mature and make large polyploid megakaryocytes. In a final step of megakaryocyte development or maturation, it will release platelets. So I'm going to talk more about details in this, about this second and third step in my next slide. What is important maybe to emphasize is that mature megakaryocytes, really big, huge cells, are being found in the vicinity of sinusoidal vessels in the bone marrow, so in so-called vascular niche, while less differentiated cells, megakaryocytes and stem cells, are present more close to osteoblast niche. So what it means when megakaryocytes matures. What happens first is that it undergoes a process of endomitosis, which means that DNA replicates, and finally what we have is a cell with up to 64 or even 128 copies of DNA. So DNA is replicating, but cell is not dividing. Another hallmark, very important hallmark of megakaryocyte maturation is formation of so-called demarcation membrane system that you can appreciate here by electron microscopy. This is a part of cytoplasm of megakaryocytes. So you can see here megakaryocyte is like a Swiss cheese. These are holes, and these are all membranes inside, which represent reservoirs for next platelets that will be formed. So mature megakaryocyte, after protein lipid synthesis and granular formation that will be transferred to platelets, they start to make pseudopods, thick pseudopods, and finally from this stage megakaryocyte is turned into proplatelet. So it's not a simple process, so this is intermediate stage. So these are represented here by staining with beta tubulin. So cytoplasm is being extended with platelet size beads along its side with branches, and finally on the ends or the tips of these proplatelets, preplatelets will be released. Preplatelets, which means that are larger platelets by size, that are around 6 microns, and this intermediates can form barbell-shaped proplatelets, which are able to twist and under fission break into 2 mature platelets. So these last steps have been realized in recent years, and these are kind of a newer intermediates that have been recognized in the formation of platelets. So I will show you how it actually happens. So this is the first video ever that shows mouse megakaryocyte turning, whole cytoplasm is being turned into mass of proplatelets. Finally, in the end, platelets are being released. And here, Okay, so here in this part, this is nuclei that will be extruded in the bone marrow. It will be removed by macrophages. Many investigations have been have found these proplatelet projections in a bone marrow sinusoid. So this is scanning electron microscopy that many years ago have observed the proplatelets extending through the junctions of endothelial cells in the vascular lumen of vessels. So what are the major motors that drive this process? So here is a proplatelet stained with microtubules in green and actin in red. So on the tips of the proplatelets, as you can see, there is a microtubule bundle. And actually, microtubules are the ones that drive this process of elongation of proplatelets, but not only just by polymerizing new proplatelets. The process that is important here is the sliding of two microtubules along each other that will push membrane and increase the size of the proplatelets. On the other hand, actin cytoskeleton in this process is very important because it does branches. So when a proplatelet needs to branch, then actin comes along, and this is a way how to amplify number of proplatelet ends. But what about other proteins? And here I'm going to talk about Filamin A. But first, I want to go back to platelets to show you the cytoskeleton of platelets. So since platelets have this disc shape that needs to be preserved in circulation, how is this maintained? So below plasma membrane, there is a spectrin-based membrane skeleton, below which is F-actin cytoskeleton. And every platelet is surrounded by a microtubular coil. So this gives a very nice and interesting ring stainings when you stain platelets for beta-tubulin. So how it looks from the cytoplasmic side. So this is a membrane, and these are F-actin polymers. F-actin polymers are interconnected below membrane with spectrin tetramers, and F-actin bundles are cross-linked with important protein Filamin A, here shown in red. So Filamin A is a huge protein. It's 280 kilodalton proteins that makes a homodimer through its C terminal domain. It has actin binding domain. It has two hinges, and the major function, how it was found, it actually was my mentor from a postdoctoral studies found it when isolating actin from rabbit muscles, and there was always this sticky protein coming out with actin together. So major function is that it cross-links actin polymers, making it three-dimensional structure of F-actin. However, this protein is very important because it also anchors membrane proteins down to the cytoskeleton, and it also serves as a scaffold for signaling molecules. So Filamin A gene is located on the X chromosome in humans and mice. In humans, mutations in this gene cause periventricular heterotopia, which is a disorder that causes malformations in central nervous system, in guts, and cardiovascular malformations. And then there is another spectrum of disorders called autopalatodigital syndrome, which affects skeletal skeleton. So it depends on the type of the mutation, if it's going to be this one or this one. And in some patients with periventricular heterotopia, there have been some descriptions of a low platelet counts and of a hemorrhage and thrombocytopenia. So when researchers tried to make knockout mice, it was embryonic lethal because of pericardial and visceral hemorrhage around 14th day of embryonic development. So Filamin A in platelets, there are actually two isoforms, Filamin A and B. Filamin A is the most abundant, and Filamin A is the one that actually links one of the most important receptors on platelets, which is von Willebrand factor receptor to underlying cytoskeleton. It links integrins and also Sig kinase. So I'm going to concentrate a bit more details on this receptor, which is actually a very complex receptor. It is composed of several subunits, GP1B alpha, beta, and 9, but the most important thing is this alpha subunit that is actually linking Filamin A with cytoskeleton. So this connection of von Willebrand factor receptor and Filamin A is important for platelet shape and circulation that I will show you in my next slides. And here's electron microscopy stainings of von Willebrand factor receptor. So every dot is one receptor, and if Filamin A is present, these receptors are aligned in these linear arrays. So this receptor is important also because there are human disease with mutations in GP1B alpha and beta subunits, which called Bernard-Soulier syndrome, which cause thrombocytopenia with the giant platelets. So we will see also that if we don't have Filamin A, it will cause similar phenotype. So since knockouts for Filamin A are embryonic lethal, we used conditional knockout with a deletion of Filamin A only in megakaryocytic platelet lineage using platelet factor IV Cre Filamin A LOX-P system. So when we analyzed these mice, so they were looking quite normal and nothing obvious. However, platelet counts in these animals were really decreased. When we looked at the bleeding time, so these animals, if they were cut, their tails were cut a bit, they were bleeding for more than 10 minutes as compared to control animals. So we looked at the expression of all subunits of von Willebrand factor receptor. We could observe decreased expression of all subunits, but not integrin. This is integrin beta 3. Also on Western blot for GP1B alpha and some degradation products. So we next checked if this receptor or GP1B alpha is connected to underlying cytoskeleton. So how we do this? We do ultracentrifugation experiments in which we pull down cytoskeleton, and these are pelleted fraction of our lysates. So if this is okay, so in control animals we see distribution of our receptor kind of equally in unbound to cytoskeleton and bound to the cytoskeleton. However, in Filamin A null platelets, there is nothing bound to the underlying cytoskeleton. Also, maybe here is not so clear, but if there is no Filamin A, GP1B alpha or von Willebrand factor receptor is randomly scattered on the surface of platelets. So next, what we looked is the activation of platelets, how they work if we stimulate them with the classical agonists of platelet signaling pathways, so with thrombin or collagen mimetic. And activation of platelets you can easily monitor by flow cytometry if we looked increased expression of P-selectin on its surface or integrin activation. So upon activation, integrins on platelets change conformation, and we can easily monitor by antibodies that recognize this different conformation. So Filamin A null platelets, they fail to completely be activated with thrombin, which is really strong platelet agonist, and also if we look integrin activation. But this was extremely true if we would stimulate platelets with collagen mimetic, both for integrin or P-selectin activation. So when we were looking at the cytoskeleton of resting platelets or activated platelets, the differences were also obvious. So in knockout platelets, there are definitely less of F-actin polymers, and knockout platelets, they fail to spread as wild-type platelets, and they don't make this lamellipodia. So there is increase in actin assembly, as can be seen here, because there's decrease in incorporation of fluorescent phalloidin after platelet are being activated. So next what we were then, so finally, until this point, we understand that platelets, their function is incomplete. Their counts are really low. So what is happening? We next looked if these mice are producing in a normal way megakaryocytes. So we looked in hematopoietic organs in mice, which are spleen and bone marrow, and we could observe increased megakaryopoiesis in spleen, both in spleen and in bone marrow, in mice which are Filamin A, LOXP, PF4, CRE. Next, we wanted to study in vitro megakaryocytes differentiation into proplatelets and platelets, and to do this, we use a model in which we use fetal liver cells from mice embryos that are 14 days old. So we mechanically disrupt whole liver and culture it in the presence of thrombopoietin for 4 or 5 days. In the culture, then you can observe big cells appearing, megakaryocytes, which then make proplatelets. And you can easily monitor morphology of proplatelets, or we can monitor ploidity of megakaryocytes. So it increases with increased numbers in a culture. Also, there's increase, of course, in a megakaryocyte marker. And okay, so if we look also Filamin A or GP1B alpha, as megakaryocytes develop in culture, they are more mature, there is a higher expression of both Filamin A or GP1B alpha. So Filamin A null megakaryocytes, they were able to make proplatelets. However, they made it less than control megakaryocytes. However, what was interesting, at the same time, we measured released platelets in a culture, and in this case, if there is no Filamin A, there was increase in platelet-like particles or platelets in a culture. So what was happening? Next, what we did was real-time videos to monitor really through every time point how megakaryocytes are making proplatelets. And after 18 hours, what we observed that megakaryocytes without Filamin A, they release platelets faster. They just simply kind of fall apart, whereas in control samples, these megakaryocytes are still carrying proplatelets on its surfaces. So I've told you, or it was quite obvious that if there is no Filamin A, these platelets were quite larger than control platelets, both in vivo, if we bleed mice, or platelets that are made in vitro. But another interesting feature was that Filamin A null platelets were stained higher for beta tubulin. So then we next examined why is this so. So what we did is we did negative stainings of microtubules. So here's every dot or little circle represents one microtubule. But what we have observed that if there is no Filamin A, platelets have more coils. So this is like a yarn. It has several rounds of microtubules around itself. And what we think that this is probably to keep their shape or to keep them more stable, because not having Filamin A has something to do with the membrane stability, which probably is true. If I show you our next slide, what we did, it was kind of spontaneous or a sporadic observation, but it gave us a clue. So if we would isolate platelets for control or Filamin A null animals, and we would keep them at 37 degrees for 24 hours, what we started to observe that Filamin A null platelets, they kind of microvesiculate, they fall apart. This process was inhibited, at least partially, if we would use inhibitors of metalloproteinases and of calpain. So when we went back and looked at the expression of some of the metalloproteinases, we saw increase in metalloproteinase 9 and in calpain 1 that were probably responsible for not only microvesiculation but also for degradation of GP1B alpha that we saw that is decreased. Okay, next step to prove what is happening in circulation was a transfusion experiment in which we isolate platelets from wild type or knockout, conditionally knockout mice. We would label them fluorescently and then we would transfuse them back into wild type animals in order to see how long these platelets circulate. So in the case of Filamin A null platelets, you can see that approximately within 2 hours more than 60% of platelets are being removed from circulation, which means body recognizes them, they are not correct, and they are being cleared fast from circulation, and that this is probably the consequence of low platelet counts in the animals. So as a summary of this first part, what we have shown that megakaryocytes make proplatelets, but this is accelerated platelet formation. They're releasing it fast in the circulation. These released platelets microvesiculate, and they're being removed from circulation by macrophages. This I haven't shown here, but we've shown that in paper. So we did also rescue experiment. If we remove macrophages, we managed to increase platelet numbers in circulation. Okay, so what about next level of controlling megakaryopoiesis? Filamin A also seems to control TPO receptor. So thrombopoietin or CMPL receptor is a homodimer. It has 2 domains. So upon binding of thrombopoietin, 2 cytoplasmic domains come closer, which activates JAK2, which is being autophosphorylated, and it phosphorylates tyrosine residues within CMPL receptor. So this triggers signaling cascade to activate STATs 3 or 5 and activates PI3 kinase pathway and also ERK kinase, and all these signals are being transferred to the nucleus and drive transcription towards differentiation of megakaryocytic lineage. But this receptor is not only important for differentiation of megakaryocytes and platelets, it is also important for survival and expansion of hematopoietic stem cells. So if we look closer to the cytoplasmic domain of this receptor, besides this membrane proximal box 1 and box 2 domains that are important for the major part of functioning of this receptor, this last 10 amino acids in the cytoplasmic domain contain sequence that resemble possible binding site for Filamin A that has been found in other known interacting partners of Filamin A. In our final experiment, we wanted to see how in vivo, what happens if mice are in need of high platelet numbers. So in this experiment, we induced immune thrombocytopenia. So we treated mice with rabbit anti-mouse platelet serum, which removes all platelets, virtually all platelets from circulation. And then we were looking at the kinetics, how platelets come back into circulation. So what we could observe is that in Filamin A LOXP PF4 mice, platelets come back faster. So kinetics is faster, and they are ready to respond faster to increase platelet counts in circulation. And another important sign of increased megakaryopoiesis or young platelets is that these platelets that are being released from Filamin A LOXP PF4 mice, they are rich in mRNA, or these are so-called reticulated platelets. So there's a stain that stains specifically mRNA, and in this case you can see the percentage of your young platelets in circulation. So as a summary or a model that we have at the moment, so if there is no Filamin A, it seems like Filamin A acts as a brake to inhibit overall or too much of activation of TPO signaling, of activation of JAK and STATs. But it seems that it's not only as scaffold, or that it is only useful to cross-link receptor with actin cytoskeleton. We think that it maybe plays a role in trafficking, that in some way it keeps it away from degradation, or keeps it away from lysosomes or proteasomes, and that is maybe important for recycling, and this is what we are trying to understand. So most of this work was done during my postdoc at Brigham and Women's Hospital, Harvard Medical School, where I want to thank my mentor John Harting and colleague Hervé Fallet the most. Now I'm trying still to establish my own lab at the Department of Biotechnology in University of Rijeka. So I have my first students there, and I need to acknowledge, of course, my funding at the moment, and also future. I just was awarded with ICGEB starting grant for another direction in the deciphering role of certain lipids in megakaryocytes maturation and progression to platelets. So thank you for your attention, and if you have any questions,